Researchers at the University of Wyoming have discovered exciting potential uses for proteins found in microscopic creatures called tardigrades, also known as water bears. These incredibly resilient animals can survive extreme conditions by entering a hibernation-like state called biostasis. The new study shows that tardigrade proteins can be introduced into human cells, causing them to form gels and slow down their metabolism, mimicking the tardigrade's biostasis abilities. This opens doors for future technologies focused on slowing down the aging process and improving long-term storage of human cells, like stem cells, which could be crucial for developing new medical treatments. Importantly, the process seems reversible, with the human cells returning to normal function once the stress is removed. This research builds on previous findings from the same team, suggesting tardigrade proteins might hold the key to making life-saving treatments available in areas without refrigeration and improving the storage of cell-based therapies. Tardigrades are known for their ability to survive extreme environments, including desiccation, drying out. In this state, they enter a state of biostasis, which is a slowing down of their metabolism. The article explores the mechanisms by which tardigrades achieve biostasis. Tardigrades use a special type of protein called CARS, cytoplasmic abundant heat soluble, to protect themselves from desiccation. These proteins are intrinsically disordered, meaning they lack a fixed structure. When tardigrades are exposed to drying conditions, the CARS proteins condense into a gel. This gel formation is thought to protect the tardigrade cells from damage. The researchers in this study focused on a specific CARS protein called CARS-D. They found that CARS-D can form a gel in vitro, and that this gel formation is necessary for the protein to function. They also found that CARS-D can condense in vivo, inside the cells of the tardigrade. This condensation appears to be reversible, as the CARS-D decondensed when the tardigrade was rehydrated. The researchers also investigated the link between CARS-D condensation and tardigrade metabolism. They found that both condensing and non-condensing forms of CARS-D could promote cellular survival during desiccation. However, only the condensed form of CARS-D helped to minimize changes in cell volume and slow metabolism. This suggests that condensation is an important part of how tardigrades enter a state of biostasis. Overall, this study provides new insights into how tardigrades use CARS proteins to survive desiccation. The researchers found that CARS-D can condense into a gel, and that this condensation is necessary for the protein to protect cells from damage. They also found that condensation is linked to the slowing down of metabolism that occurs during biostasis.